DNA replication, copying the genetic code. So the first step to this is called initiation, and we know that the replication of DNA is a semi-conservative process, meaning that we've got this double-stranded DNA. It's going to split apart, and then each part is going to be copied, so you end up with two versions that are half the original version and half of a new version. So the DNA is first going to be unwound, and the enzyme that does this, each has got its own name in it. So this particular enzyme is called helicase, and this is the one that unwinds the DNA. And it's not going to happen necessarily just in one spot, so we can have this opening up in several spots. You can see here's one particular spot, and here's another spot, and here's another spot. And so the replication is going to be moving in a particular direction. So if you looked at like this little circle in the middle here, it's going to open up that way and it's going to be opening up that way. So eventually, as both sides open up, plus these sides are moving as well, and they're going to meet, and eventually you're going to end up with two new double strands of DNA, and then each cell can get one copy of those and be a copy of the original. So there are multiple places where this takes place, multiple origins for the actual replication bubble to take place, and then these things on either side of the bubble are called replication forks, and they move in opposite directions. So there is an enzyme called Gyrase that is going to be moving ahead of this area of replication, and it's going to, because imagine if you had a rope that was twisted and you started pulling it apart, it's going to want to super twist in the upstream of where you're actually pulling it apart from. And so this enzyme Gyrase here is actually going to relieve that tension by essentially breaking the DNA and allowing it to unspin and then reconnect again. So imagine this was one strand of DNA, or one side of the strand of DNA, and this is the other side on the bottom here. And so here is the double helix part. So this would be the bubble that's opening up. The replication fork, the way that it's opening, is going to be moving in this direction here on this side of the bubble, and the opposite direction on the other side. Upstream here, this is where the Gyrase is going to be doing its work. As the double-stranded DNA opens up, it's going to create a lot of tension up in this upstream area, and so Gyrase is going to be able to relieve that. There are also these single-stranded binding proteins, because the two strands, single strands of this DNA that make up the double-stranded helix, they're going to want to come back together due to hydrogen bonding. So single-stranded binding proteins help to keep them apart, so that these machines here that are going to, so here's your helicase fork, once it's gone past, to keep this end here and the end up here from rejoining, we have these single-stranded binding proteins. So these other machines that are going about the copying process can do their work. So this elongation part, this is the building complementary strands. So opening up the strands, that's one thing. Now each of those strands is going to have to be copied. So remember, the DNA itself is described as anti-parallel. So the DNA strand has a particular end to it. It has a 3' end and a 5' end. The 3' is the one with the OH, because remember, it's a sugar phosphate backbone. And so you have this sugar, and it's attached to this phosphate group up here, and then we have this nitrogenous base that sticks off into the middle, and so that's going to make up your one nucleotide. And so we have the 3' end down here with the OH on it, and we have this phosphate, that's a P phosphate group up here, and so this is the 3' end. So if you think of the bottom side of this nucleotide here, and then we have this 5' end up at the top here, the phosphate end. So if it's a sugar phosphate backbone of alternating sugar phosphates, which makes up a one nucleotide, and then of course this would be attached to a phosphate group of another nucleotide, pretty messy nucleotides, which has its own nitrogenous base attached to it, and again it has this OH group on it here, and so this would be the 3' end. So no matter how long it is, it's going to have one end that's a 3' and one end that's a 5'. Now that is one direction there, and then the other DNA that has the other nitrogenous bases, hydrogen bonded, right? It's going to also be attached to a sugar phosphate backbone, but this one's running in the opposite direction. So this is a, I'm going to draw upside down P. So this is the sugar phosphate backbone, so that would be one nucleotide there, and again the nitrogenous base pointing on the inside, and so it would have an OH group over here, and that would be the 3' end. And then of course this would be bonded to another phosphate that's connected to that nitrogenous base that I have in the middle here. Sorry, another sugar phosphate group. And so there's my other upside down P. And so again, this is the 5' end. So these are running anti-parallel to each other, and the hydrogen bonding between the nitrogenous bases in the middle. OH group is the 3', phosphate group is the 5'. So these are anti-parallel, and that's a much better picture than mine, but you can get the idea here. Sugar, let's get this a little bit more bolded. So there is a nucleotide, and it is bonded to this other nucleotide. So there are two nucleotides, and again, this is the 3' end, and that is the 5' end, and the opposite on the other strand. So this DNA is built using these nucleoside triphosphates. That should remind you of something there. And so here we have these 3 phosphates as it's this individual monomer, and as it gets attached onto the growing chain, this OH group here at the end is going to attach to this phosphate group here as these phosphates get broken off, and then we get this bonding happening between the OH and the phosphate group here. And so this becomes attached now onto the previous, so this is the old chain here, this is this part right here, and again this nucleoside triphosphate is going to lose 2 of its phosphate groups and keep one of them and get attached onto the growing chain, and it is going to be growing in this direction here, so it's always constantly being attached onto the OH of the old or already existing chain. So the next phosphate to be attached would be attached to that OH group there. Primase is the next sort of machine that comes on the enzyme that comes onto the scene here, and essentially what it does is it creates an RNA primer. And so this stuff is not made of DNA, which is odd that we're trying to grow DNA here, but it actually uses RNA to start the new chain. That's why it's called a primer. So it has a little length of them, so like 10 to 60 base pairs ish. It'll lay down this primer, and then DNA polymerase 3 is able to use that primer to start building the new chain. So imagine, again, we've got this replication fork here, and we should be able to picture it from either side, but the double-stranded DNA has been opened up by helicase, and so now we have these single strands of DNA. We've got the one on the top here and one on the bottom here that are going to be copied. They're going to be matched up with their base pairs, and then we're going to have 2 new double-stranded DNAs. But in order to get that done, Primase is going to put this RNA primer here, so let's do that in red here. And so this chunk right here, you can imagine that being 10 to 60 bases long. They'll match up with the nucleotides that are already on this previously existing DNA right here, and it'll match them up here. And again, this RNA primer also has a 5' to 3' end, and so this 3' end right here has an OH group on it. That's what makes it the 3' end. So just as we saw before, that's where a new nucleotide is going to be able to be bonded on there. But it starts with this RNA primer. So DNA polymerase 3 uses this primer and builds on it, and it is going to attach only to the 3' end. That OH group of the primer is where that first nucleotide is going to be laid down, and then the next one can go on it, attached to the 3' end, and then the next one, and the next one, and the next one. So it needs this primer because it can't just start. DNA polymerase 3 has some limited abilities. It can only add nucleotides to a 3' end, a hydroxyl group that's already there. So imagine this is, again, looking at it from another direction here. Notice the 5' end here. So this is a 3' end. So this last nucleotide has an OH group hanging off there, and that is what DNA polymerase 3 is able to work on. So it can then take this nucleoside triphosphate, make sure I get the right part here, nucleoside triphosphate, and it will be able to attach this phosphate group to this OH group, and therefore, and notice this is an A here, so it's going to have to lay down a T. It'll be able to put this one right here, and it'll be able to bond it to the previously existing nucleotide that is right there. That nucleotide has to be there in order for DNA polymerase 3 to be able to attach the next nucleotide, and therefore you need this RNA primer there to start off the chain. So synthesis, the building of the strand, can only occur in the 5' to 3' direction. That's how DNA gets built, and it happens fast. So since the daughter strand, the new strand that's being formed, can only be built in this direction, 5' to 3', only adding onto the OH end of it, there is going to be one of these strands, because remember these strands are running anti-parallel. So if you have this double-stranded DNA and then you split it open, one of them will be 5' here and 3' here, And then the other one is going to be 3' and 5' as they are anti-parallel. So that means that when you go to add on a primer onto it, so let's do this in green, so this new daughter strand is going to be laid down. It'll have a 5' end, which means the other end here is 3'. So that means this end can be built off of, and as the replication fork opens up further, this new strand in green is going to be able to continuously be built. So this is called the leading strand because it is able to move continuously as this fork opens up. So you can imagine this is going to get further opened up, and so as the replication forks moves in the way I've drawn it here to the left, this leading strand is able to continually, let me add on here, so continually add in this direction towards the replication fork. However, let's do this in blue. If we put a primer here, because its 5' end is here and its 3' end is here, it can only be built in the direction going away from the fork. So it's going to have to be done in these, it's called the lagging strand, and it's going to have to be built in this sort of piecemeal fashion. We're going to build a little bit, and it'll open up a little bit more, then we can build a little bit, and we can open up a bit more, and so on. So it is non-continuous. So think our leading strand is synthesized towards the replication fork. So if the replication fork is moving in this direction here, then, and let's try to place our leading strand on this particular one. And again, we can look at this from any angle we want. This is why we put the labels on. So since this new strand here has the 5' end on that end and the 3' on this end, it is going to be the leading strand. On the other end, let's do this in green here, we're going to have the lagging, let me put this up here, lagging strand. So this, it's going to be able to build one here, which will have a 5' end and a 3' end. Now remember, the fork is moving this way, but DNA can only be built from the 3' end. So it's going to be called the lagging strand on the bottom here. It is going to be built, it'll end up being the same thing, but it is called the lagging strand because as it gets built, it has to be built away from the replication fork, and so it has to be built one piece at a time, as opposed to the leading strand, which is built in the direction of the movement of the replication fork and therefore can be built continuously. So the lagging strand is going to be built up of these segments called Okazaki fragments, one piece at a time, because it's moving in the opposite direction of the replication fork. So let's look at this picture here, and again, each time you look at it, they can be drawn in any number of ways. It can be moving to the left or to the right, and the lagging / leading could be on the top or the bottom, and so we've got to put the labels on it and figure which way it's going. So this particular one, we can see the replication forks moving to the right here. If we look at the top strand that is being copied, the yellow part here is the primer and has a 5' end, which means the 3' end of the primer would be there, which means DNA polymerase 3 is able to add on nucleotides and move in that direction, the same direction as the replication fork. So that is the leading strand. It moves in the same direction as the replication fork. And there's no set direction, right? It could be moving left or right. It depends on how you draw it. Up, down, backward, doesn't matter. So you've got to be able to figure out which end is which with the labels. The lagging strand, that's the more complicated one, that's what's happening on the bottom here. So the RNA primer will be put on. You can see the 5' end is there. So this would be the 5, let's do this in red. This would be the 5' end of the primer, which means this is the 3' end, which means DNA polymerase 3 is going to start adding on to the 3' end, and it will move in that direction, which is fine, but you'll see that it's not in the same direction as the replication fork. So it is going to have to be built a piece at a time, because eventually it gets to the end, and that's it, it can't do anymore. Meanwhile, as it's gotten to the end, more of the DNA here has been opened up. You can see the single-stranded binding proteins holding it open. And so while the leading strand just keeps going, the lagging strand has to be restarted. Each segment that you open up, you're going to have to get a new primer. So there's going to have to be a new primer set right here. And then it's going to have a 3' end there, right, to match up. And so then it can be built in the only direction that DNA polymerase 3 can move is on the 3' to 5' end. So it'll be able to move all the way till it gets to that first primer that was laid down there. And then the DNA will be further opened up, and the process continues over and over and over again. And so these fragments here that consist of a RNA primer plus a segment of DNA, these are called Okazaki fragments, and they are built because the lagging strand can only be built in the 5' to 3' direction, and therefore it's moving against the direction of the, or not with the direction of the replication fork. So once DNA polymerase III reaches these upstream primers, so as it goes through, it's eventually going to hit those primers. It'll stop laying down new nucleotides, and there is another DNA polymerase I, which will take off the RNA primer, and it will replace those RNAs. So and again, it's moving in that direction. It's able to do this in the 5' to 3' direction. It'll remove those RNA nucleotides and replace them with DNA nucleotides. However, because it'll get to the end here of the primer where the actual DNA starts, it'll get to the end and then it's got no more RNA to remove, and so it'll just stop doing its work there. So what you'll find is that there'll be a gap between the two nucleotides. That phosphodiester bond that is building the sugar phosphate backbone will be not connected, where the original primer and the original DNA segment, the middle of the Okazaki fragment, where they met. And so then DNA ligase is going to come in and build that particular, or bond those particular nucleotides together, and that is going to link the sugar phosphate backbone together. So we will eventually have one long continuous piece of new DNA. So here's the overall view of it. So we have helicase unwinding the double helix. We have the single-stranded binding proteins keeping it unwound, so they don't hydrogen bond back together. The leading strand is continually synthesized, and again, you can see in this particular one here, the 5' of the new strand is on the right, and so that means the 3' is down here, and if it's moving in 5' to 3' direction, and that's the same way the fork's going, that's going to be the leading strand. The lagging strand, though, and again, for the new DNA, the 5' would be here, and so the 3' of this little primer would be right there. So DNA polymerase III is going to be able to move in this direction here, away from the replication fork. So then a new primer will have to be put down, and then on its 3' end, the DNA will be able to get built until it meets up with the previous primer. And again, we then have DNA polymerase I replacing these nucleotides here with, which used to be RNA nucleotides, that will replace them with DNA nucleotides, but right where that primer met the new piece of DNA, there's going to be a gap, and so ligase is going to have to come in and form the bond with that sugar phosphate backbone. In terms of errors, as these nucleotides are being placed down, they're always matching their complementary base pair on the opposite strand, the one that's the original strand that's being copied. So every once in a while, some things go wrong, and so this is where DNA polymerase III can actually, as it's laying down these new nucleotides, if it puts a mismatch in there, it's able to reverse and replace them if that is noticed by it, or if it's picked up with it, and again, it's not a conscious or anything like that, just it doesn't quite fit, it'll try to replace it with another one. And then we also have this other team of enzymes like DNA polymerase II, which is not on its own, but is able to actually go over afterwards and start sort of see wherever there is a mismatch, because remember, they're going to be hydrogen bonding to that nucleotide on the other side, and if they don't hydrogen bond properly, it's because it's the wrong match. And so anywhere there is this mismatch, is able to extract that nucleotide and replace it with a new one. Not just copying can cause these, but actually environmental factors as well. Things like radiation damage can cause these nucleotides to be mismatched, and then you have this error in the DNA code, and hopefully it'll get picked up by DNA polymerase II.